Okay, let's unpack this. We are taking a deep dive into, well, the ultimate biological blueprint today. We're synthesizing a stack of really cutting-edge research to answer one massive question. How exactly does science predict your biological destiny?
It's a huge question, and it really is a journey into prediction. We've pulled together sources on everything from, the specific enzymes that let some people live past 100
Right.
to these enormous genomic projects that are mapping extinction risk for, like, every mammal on Earth. And then there's AI, which is it's completely changing the game.
So our mission today is to give you a clear, structured understanding of the molecular rules that govern life and death. We're pulling from molecular biology, from these big human aging trials, and the incredible power of genomics.
Right.
If you want a shortcut to understanding the precise mechanisms driving your health span, this is it. Let's jump straight in. The molecular secrets of long life. We're starting with a remarkable enzyme. It's called SIRT6.
SIRT6, yeah. It's a class of proteins that are just crucial for metabolism, cellular health, that kind of thing.
Researchers, they were trying to find the key to, extreme longevity. So they sequenced the SIRT6 gene in a very specific population.
A group of 450 Ashkenazi Jewish centenarians.
People living past 100. And they compared them to hundreds of controls and found this rare variant that was way more common in the centenarians.
Right. They called it sense-IRT6. It's defined by just 2 little amino acid substitutions. But what's so fascinating here, and it really goes against what you might think about a superprotein, is the surprising trade-off in what it actually does.
Okay, here's where it gets really interesting for me. Because you'd assume a longevity enzyme is just an accelerator, right? It does its job faster, better, all around.
That's a logical assumption.
But the sense-IRT6 variant, it actually has a reduced deacetylase activity. Wait, so you're telling me the best version of this enzyme is actually less good at one of its main known jobs?
Precisely. That's the nuance that the field is, still wrapping its head around. While that deacetylase function is lower, the centenarian variant has an enhanced activity in its other main role, which is mono-ADP ribosyl transferase, or AMAPK. It's that specific shift, that trade-off that seems to unlock the benefit.
So it's about prioritizing one job over the other. And how does that specific enhanced main of ADPR activity translate into a longer life?
It all comes down to superior genome maintenance. And that really is the cornerstone of longevity.
So stabilizing your DNA.
Exactly. We're talking stronger suppression of these things called line 1 retrotransposons. You can think of them as jumping genes that destabilize your genome over time.
Oh, it holds them in place.
It holds them in place. And critically, it also led to enhanced repair of DNA double-strand breaks. And on top of that, showed a much more robust ability to kill cancer cells compared to the common SRT6 variant.
And the researchers could actually trace this back to a physical interaction, right?
Absolutely. The enhanced MADPR function, it leads to a stronger physical bond with Lamin A, or LMNA.
Which is, what, like a scaffolding protein?
It's the scaffolding in the nucleus, yeah. And that interaction is key. It provides a real structural explanation for how this variant keeps the genome more stable. The whole point is that longevity isn't always about turning the volume up. Sometimes it's about turning the right dial up.
Speaking of dials, let's shift from genetics to lifestyle. Specifically, the landmark calorie trial. This studied the long-term effects of caloric restriction, CR, in people.
Right. This was designed to see if consistently eating about 15% fewer calories actually slows down biological aging. And they used these incredibly precise DNA methylation clocks to measure it.
The thing is, the initial data was genuinely confusing.
Yeah.
Because they used 3 different ways to measure biological age.
Yeah, you had 2 of them. Phenoage and grimmage, which are designed to predict your static accumulated risk. They essentially give you your current biological age.
Okay.
But the third, and this is the critical one, was the DunedinPACE clock, and that measures the dynamic pace or the rate of your aging.
And if you only looked at this first 2, the static clocks, the results were kind of disappointing. The control group and the calorie restriction group, they didn't really differ on phenoage or grimmage scores.
Right, so people were asking, if you didn't reduce your accumulated biological risk score, did the diet even help?
This gets at a really essential distinction that you, as a listener, need to grasp.
Phenoage and grimmage are kind of like the odometer on your car. They tell you how much mileage, how much wear and tear you've accumulated. CR didn't dramatically roll back the odometer.
But DunedinPACE,
Okay.
That's the speedometer.
Exactly. It measures the rate at which you're accumulating damage. And that was the breakthrough finding here. CR did significantly slow the pace of aging. as measured by DunedinPACE.
At both 12 and 24 months.
Yes, the effect size was very clear. So caloric restriction makes your biological clock tick noticeably slower, even if the years of accumulated damage aren't, instantly erased. It shows we can actually change the speed of aging.
Which is a whole new level of biological control. And that sets us up perfectly for our next segment, quantifying health span and functional resilience. The concept here is immune resilience, or IR, and the survival gap it creates is, honestly, it's stunning.
Immune resilience is what's called a salutogenic force.
Yeah.
It's a fancy word, but it just means it promotes health rather than just fighting disease.
Okay.
And optimal IR is this very delicate balance. You have high immunocompetence. Your body is great at fighting threats, while at the same time keeping inflammation low. It's kind of the perfect immune status.
Let's focus on the payoff for that, because the numbers here are just staggering. A 40-year-old with poor IR has a mortality risk that's 9.7 times higher.
Right, and that risk is equivalent to a 55-and-a-half-year-old who has optimal IR.
So we are looking at a 15.5-year gap in expected survival, just from the balance of your immune system.
A 50-and-a-half-year gap, yeah. And the ability to maintain that state, especially when you're under stress, seems to be controlled by one key player. Transcription factor 7, or TCF7. Elevated TCF7 expression is what sustains that optimal immune resilience.
So how does that play out in real life, like when you get really sick?
Well, the researchers found that TCF7 levels were highest in this group they called IR preservers.
Okay.
These were people who, even when they got a severe respiratory infection, think influenza, or even COVID-19, they kept their optimal immune status. They fought off the infection without the runaway inflammation that causes so much damage.
So TCF7 is like the switch that prevents that inflammatory cascade.
It appears to be, yeah. A prime target for future interventions.
But the research also found a pretty crucial age limit on this whole protective mechanism, didn't it?
It did, absolutely. It's almost like a biological warranty period. The benefits are tremendous in midlife, but the protective power of optimal IR, it really wanes after age 70.
So the mortality rates start to converge.
They do. They start to converge between the resilient and non-resilient groups. It suggests that age-related issues eventually just become too overwhelming, regardless of how good your immune system was before.
That convergence point, age 70, is so important for timing your health strategy. We'll come back to that. But let's pivot to another measure of functional ability, intrinsic capacity, or IC.
Right, IC. It's a measure across 5 domains: psychological, vitality, sensory cognition, and locomotion.
And the data here showed this fascinating split in how men and women experience functional decline.
A literal timeline of vulnerability. It was very clear. Males had significantly higher scores in the psychological and vitality domains.
Okay.
But females scored higher in the sensory domain.
But the real insight is the timing. The data shows that women exhibit a much earlier sensory decline, starting pretty dramatically around age 42. For men, it was age 67.
So that's vision, hearing, balance. Women are, on average, losing those abilities much sooner.
And interestingly, the complete reverse happens with cognitive capacity.
That's right. Men showed earlier cognitive decline, starting around age 72. Women tended to maintain cognitive resilience for way longer, with that decline not starting until around age 86.
So you have two totally distinct trajectories. It really highlights that aging isn't just one thing. It's a collection of very specific declines. Okay, let's zoom out now. From individual humans to, well, the fate of the entire mammalian family tree. And this is thanks to the Zoonomia project. This thing is just planetary in scale.
Oh, it's staggering. This project generated genome assemblies for 131 species that had never been properly characterized.
131 new ones.
Which resulted in a whole genome alignment of 240 eutherian mammals. That's over 80% of all mammalian families. It's like creating the ultimate Rosetta Stone for mammalian genetics.
And the immediate practical application for biodiversity conservation is what makes this so important. How does sequencing a bunch of animal genomes actually help save endangered species?
They found that these regions of reduced genetic diversity, they're called segments of homozygosity, or SOH, are much more common in species facing a high risk of extinction.
SOH basically indicates inbreeding.
Inbreeding and a loss of the genetic flexibility you need to adapt. It means conservationists can perform a kind of biological triage.
Right. Sequencing just one individual from a species, you can get these critical diversity metrics like SOH and identify which populations are at imminent risk. You can prioritize your resources.
And they focus on some really critically endangered species, like the northern white rhino.
The northern white and black rhinoceroses, the Russian saiga, the Mexican howler monkey. Yeah, the list goes on.
a direct conservation tool. But this comparative genomics work also had this massive medical utility, especially during the COVID-19 pandemic.
It did, yes. By comparing the structure of the ACE2 receptor, which is the doorway the virus uses to get into cells, across all 240 mammals, they could predict vulnerability.
So they could see which animals were most likely to get it.
Exactly. The analysis immediately flagged 47 mammals with a high or very high likelihood of being SARS-CoV-2 reservoirs or hosts. That's the power of having a truly phylogenetic scale model.
And that ability to predict outcomes brings us perfectly to our final segment, how AI is now becoming the ultimate prediction tool in genetics, specifically with something called variant effect prediction, or VEP. The challenge here is just immense. The human genome has millions of variants, and we desperately need to know which ones are harmless and which ones cause disease.
And the traditional methods, they kind of struggle with that.
They do, because they rely on homology. They need to compare a new variant against known similar sequences across evolution. If you have a brand new variant or one in a poorly studied gene, they hit a wall.
And that's where the protein language model comes in. We're talking about AI derived from natural language processing, the same kind of tech that generates human text.
That's right, the ESM1B model. It's this massive, deep neural network, and it was trained on 250 million protein sequences. The language it's learning is the sequence of amino acids that makes a protein function.
So it's learning the grammar of life.
That's a perfect way to put it. It learns what sequences are highly likely to fold and work correctly, and which sequences are just biological nonsense.
So if I introduce a misspelling, a genetic variant, the model can tell me if that results in a working protein or a broken one without a lab experiment.
Precisely. The huge advantage is it doesn't need that explicit homology. It can estimate the likelihood of any possible amino acid sequence, so it can score variants the older methods couldn't even touch.
And how did it perform? How did it stack up?
Extremely well. When they tested it on the big ClinVar dataset, distinguishing between pathogenic and benign variants, the AI model, ESM1B, outperformed the leading comparative method, EVE. It was just more accurate.
And crucially, this can address that huge backlog of genetic question marks, the variants of uncertain significance, or VUS.
Exactly. I mean, before, these VUS were just diagnostic dead ends. They left patients in limbo. ESM1B can assign scores not just to simple mutations, but to more complex ones, like insertions and deletions.
Wow.
Their analysis of the VUS indels in ClinVar estimated that 52%, over half of those unknown variants, are likely pathogenic. That just immediately fast-tracks diagnosis for thousands of people. It removes the uncertain label.
What an incredible synthesis. I mean, we've gone from extreme human longevity being driven by this precise molecular trade-off in RNA-seq
Right, prioritizing DNA maintenance.
to seeing how caloric restriction changes your dynamic rate of aging, your DunedinPACE, not just your odometer.
And we learned that immune resilience, that perfect balance, buys you a striking 15.5-year survival advantage. It's regulated by things like TCF7, which turned out to be critical even during viral outbreaks.
And finally, we connected that individual health to, well, global biological destiny, seeing how zoonomia provides conservation and triage for mammals, and how AI, acting as this protein language model, is poised to resolve all those variants of uncertain significance.
Bringing it all together, that immune resilience research really does highlight a biological runway for our health. And those crucial survival advantages, they decrease sharply after age 70.
Right.
Given that we can now measure the dynamic speed of your aging with DunedinPACE, and we can identify your key regulators of resilience like TCF7, you suddenly have this unprecedented visibility into your personal biological future.
So, what does knowing your individual biological runway mean for how aggressively you choose health interventions, especially in your midlife? Knowing that the protective capacity of your immune system is going to start to wane after 70, should optimizing immune resilience, managing that inflammation and competence balance become the central, most urgent health goal of your 40s and 50s? Something for you to consider as you decide which dial to turn first in your own blueprint